This is the Discovery Files podcast from the U.S. National Science Foundation. As we start a new year, we're looking back at some of last year's most popular episodes and revisiting some of the stunning research that we featured. Early in 2024, we looked at several innovations with medical applications. One project involved investigating how white blood cells interact with cancer cells. We caught up with Kolade Adebowale, a postdoctoral fellow at the Harvard John A. Paulson School of Engineering and Applied Sciences, who was working with attaching a small backpack to cells that could be used to deliver treatments to cancer cells.
As you can imagine, we have this particle that's attached to the surface of a cell. One can envision a scenario where we incorporate some drug of interest in this microparticle. This can be like a therapeutic drug, really like a cytokine. It can be whatever drug of interest to you. So that's one reason one might use this cellular backpack. But even beyond that, it appears as though the mere process of attaching this microparticle to some kinds of cells, so for example, things like natural killer cells, which are a type of white blood cell, or T cells, which are another type of white blood cell, just as physical attachment, it appears as though is able to activate these cells, which means that they have enhanced ability. It almost endows them with some kind of enhanced superpowers to be able to fight cancer specifically.
Amber Dorian, assistant professor in the Department of Electrical and Biomedical Engineering at the University of Vermont, told us about her work developing biofilm-resistant wound dressings.
Biofilms are basically a different way for bacteria to grow. So bacteria that we're all used to envisioning, we kind of think of them as floating around or swimming, in some sort of liquid, but they can also attach to a surface. And when they attach to a surface, they kind of create this really nice, happy home for themselves, this environment that's protective for them. And that surface can be something like the surface of a wound. If you have an injury and it's healing, the bacteria can attach to it and adhere to that surface and create a protective environment for themselves, which is not necessarily good for the person with that healing wound. They can also attach to surfaces of medical devices, things like catheters, heart valves, things like that are indwelling or inside the body. So biofilms present a really large problem for treatment. When the bacteria attach to that surface, they're just much harder to treat. They create kind of a slime environment for themselves to live in that makes it hard for antibiotics to get to the bacteria. It makes it hard for cells of our immune systems to get to the bacteria. Wound dressings, historically, obviously have been used for a long time, maybe just a piece of fabric, or when gauze was developed, that was used as a wound dressing for a very long period of time and still is. More advanced wound dressings have tried to create environments of that wound. Say if we're talking about a topical wound, say someone's had a burn injury or a traumatic injury, the skin is broken, and that creates a place where bacteria and other environmental factors can come into the body, creating lots of problems for that patient. And so a wound dressing creates a protective barrier between the outside of the body and the inside of the body. And it also is providing a healthy environment for healing. But they also have a high incidence of themselves becoming colonized with bacteria. So what we've tried to do is create a wound dressing that essentially is an anti-biofilm wound dressing, where it inhibits the growth of the bacteria, both on the wound dressing, and helps the wound itself avoid the bacteria as well. So we're trying to help the patient heal by preventing the wound dressing itself from becoming attached to bacteria. Our approach to this relies on eliminating what essentially is an energy source for the bacteria. And it turns out that helps to inhibit these biofilms. It actually can both prevent the biofilms from starting and treat the biofilms if they're already established. So by incorporating some of those approaches into our wound dressing, we can cut down on the bacteria, even being able to start a biofilm on the wound dressing itself, and help treat the potentially already established biofilm that's in the wound.
Dmitry Kiriev, assistant professor of biomedical engineering at the University of Massachusetts Amherst, joined to discuss developing biosensors using graphene.
It is the electronic properties, this combination of atomic distribution of the material that makes this unique sense. But then it's not only about conductivity itself, it's about sensitivity to external field, to the surroundings. I have this example. You have something hot, let's say you're cooking, right? You put your hand on and you try to sense something, you would only sense with one side of your hand, right? Because it's thick, it's not one atom. You don't sense anything with the opposite side of your hand. But if your hand was one atom thick, you would sense it, much better, faster, with a greater level of sensitivity. And that is the case with graphene and 2D materials. All of these atoms, the whole surface is exposed to the surroundings. That makes it amazing sensor.
We learned about how clocks work and why precise measurements of time are important from Eric Hudson, a professor in the Department of Physics and Astronomy at UCLA, who has spent the last 15 years working to accomplish the seemingly impossible, directly manipulating the energy level of an atomic nucleus using a laser, something that has never been done before and could unlock the most accurate clock ever.
We all have an inherent understanding of what a clock is. They're very important in our lives, but we don't really think about them too often. What they're doing is pretty profound. Evidently, there's something we all agree on called time. The time is later now than when we started this discussion. But how do we measure that? How do we agree upon how much time has passed? And what we do with clocks, we typically have some sort of process that repeats itself, and we assume that repetition is sort of constant in time. So think of a grandfather clock where you have a pendulum swinging back and forth. After one oscillation back and forth, we call that 1 second. You can let 2 oscillations be 2 seconds, 60 be a minute, and so on. The wristwatches or our phones that we use, they typically have something very similar to this. It's a piece of quartz that vibrates at about 30,000 times per second. You could see a lot of details in that analogy that are going to be important for understanding atomic clocks or eventually nuclear clocks. The other thing that's important is that our clocks be identical. If you and I build grandfather clocks, and I make my arm a little bit longer than you, or my bearings maybe not as good as you, there's a little more friction, right? Then my clock will run slower than yours. And so after some time, we no longer agree on how much time has passed. And I'll show up for this interview late, and that is where atomic clocks come in. As far as we know, every, let's say, hydrogen atom in the universe is identical. And what that means is they absorb and emit light at exactly the same frequencies, right? So we know you can shine in a laser, for example, and excite an electron in an atom to a higher orbital, and that happens at exactly a certain color of light. And it's the same here as on the moon, as in Andromeda Galaxy. If I make sure my laser can always drive that transition, now my laser, the electromagnetic field of the laser, is oscillating at a frequency that's tied to the structure of hydrogen, which is set by the laws of the universe. And so my laser now becomes my oscillator. And if I count the oscillations of the electromagnetic field, I can use that to keep time. That can be very high frequency in optical atomic clocks. It can also be identical because all the atoms are the same of the same type. So our clocks are guaranteed to tick with the same frequency. And importantly, because we have techniques now where we can isolate these atoms from their environment, we don't have this problem of friction in the pendulum clock anymore. Right. And so these things aren't going to be perturbed by their environment as much. And so we can build these, amazingly accurate clocks that people have built. Precision timekeeping is actually really important in our daily lives. We don't normally think about this. If you're a minute late to a Zoom meeting, it's okay. But your cell phones, your global positioning, lots of communication technology, some imaging technology, you really have to have the most precise clock you can get. And if you had better clocks, you could do those things better. If we had better atomic clocks, GPS would be better. That is important for, navigation here on Earth, but also as you go into the solar system and beyond, this sort of deeper space navigation, this becomes a really big problem.
Technological advances, including machine learning and artificial intelligence applications, continued to be popular subjects. We spoke with Miyoko Chu, Senior Director of Communications at the Cornell Lab of Ornithology, who was involved in developing the popular Merlin Bird ID app, which helps users to identify birds in the wild from their mobile devices.
The origin of Merlin went back to about 2009. Our team here at the Lab of Ornithology was wondering, how can we find some funding to do more to engage people in our websites about birds? So we sat down and we had heard about this NSF opportunity, a grant that could be available in informal science education. And we said, what could we do that could be valuable? And we noticed that when you look at our All About Birds website, which is like the online encyclopedia of birds, you looked at what people were typing into the search box. It was things like little brown bird with white line, red bird with a crest. So there were these descriptors where clearly people were trying to identify birds using our website as a bird identification tool. But that's not the most useful tool to help you ID birds, although if you spent a while, maybe you could figure it out. So it happened that one of the developers on our team said, I've seen this online game about 21 questions where the computer guesses what you're thinking based on clues that you provide it. And that was just at the beginning of AI really taking off. Like, I hadn't really seen applications of AI before, but we said, wow, a computer can guess what you're thinking in 21 questions? That sounds neat. Can't we do that for birds? So that was the start of the idea, the nugget of the idea. I have to say, I'm completely astounded just by how many people have started to use it. But the reaction has just been delight and a sense of, new possibilities, because as soon as people realize that they can use this app to give them the name of something that they've seen or heard, it just becomes much more tangible. Once you have the name, you can learn all kinds of interesting facts about it. So it really opens a door.
Tanya Berger-Wolf, a professor and computer scientist at The Ohio State University, joined us to share how she is using technologies including artificial intelligence to extract information from images in the new field of image genomics.
So it's the omics of images, imageomics, right? This is the field of discovering biological information from images, particularly focusing on traits and phenotypes. So traits are the things that we consider important about animals, we can observe, and we can connect to function, often through genotype. So things that started with Mendel's discovery of the size, color, shape of the peas, and the pea plant, and the pea pod, and the pea flower. And that's how we started on the pathway to genetics and then genomics. But traits can be at the individual level, so the things like color, shape, size, weight, height, the pattern of the zebra stripes. They can be at the population level, the variation of the colors and shapes, but they can also vary during an individual lifetimes. Things like behavior, or pregnant, sick, healthy, juvenile, adult have different proportions. Things like for the plants, so not only animals have traits, certainly plants as well, like peas, but the width and length of the leaf as it grows, the ratios of all this as the nutrition in the soil changes and the amount of sunlight changes, all of these are traits. And then the product of genes, environment, and the combinations of the two. Now, the traits are not just important to observe and understand, but it is important to connect them to function and the context and the understanding of why. Not only what we're seeing, but why is it changing? Why do animals have traits? What is the intra-species trait variability within the species, and how is it related to the variability in the habitat or the conditions of that species along a particular gradient? And so as humans, we're very biased by what we look at, what we can see, and what we consider important in terms of traits and everything else for that matter. There are so many things that we're not even capable of seeing because our apparatus that we're using to see is highly unreliable and is limited. For example, we don't have enough acuity to distinguish red-orange variability in polymorphic moth. because that did not evolve for our benefit. That variability evolved for birds, right? To fool the birds and to signal something to their own species, insects. Our vision is very, very different. There are also traits that are not even in the visual spectrum at all. There's chemical, the acoustic, and all the other kinds of sensory channels that we're not great at perceiving. And so we're missing a whole bunch of the world. And that translates into missing understanding of a whole bunch of the world. So where technology comes in, it makes the invisible visible. Because we're not only can bring in images from the same spectrum that we humans are seeing, but from a whole other range of spectra. And we can make sense of that. We can extract information from that and then connect it to a whole other wealth of data that then really not only finds new traits, connects them to different contexts, and starts through these patterns of connections, understand the function of these traits and how they evolve, why they evolve, and what is their function in the also environmental context, in the ecological context, and understanding the truly, maybe, the life on Earth.
We also explored an interagency effort that resulted in the Global Artificial Intelligence Research Agenda, a document crafted to serve as a starting point to align a global research vision, with Michael Littman, the Division Director of Information and Intelligence Systems in the Computer and Information Science and Engineering Directorate at the U.S. National Science Foundation.
So it's worth pointing out that the idea for the GERA was first made public in the executive order on artificial intelligence, which the Biden administration issued in last October. So we're getting really close to the 1-year anniversary of it. In that document, there was a whole slew of things that were requested of demanded of the agencies to actually do. Some were targeted at particular agencies, some were targeted to all the agencies. This particular item, it mentioned creating a global AI research agenda, so which we've been calling GERA, but it doesn't actually say GERA in the document. We now know that this is actually the name of a monster from Godzilla. So that was, foresightful. It's not a monster. It's actually a really great thing. So the idea of it is that the executive order said that the State Department and USAID agencies in the US would put together a global AI research agenda. Basically, what should we all be doing together to help move AI forward in a way that advances artificial intelligence for the benefit of the entire planet? The authors of the executive order, they felt that the State Department and USAID have the expertise to really connect a US vision to help turn it into a global vision, to really connect with our partners in other countries. They also asked the Department of Energy and the National Science Foundation to be a part of the development of the research agenda because, well, AI research really happens at these agencies within the federal government. We also ended up bringing in the Department of Labor to help out with the issues that have to do with the way that AI is being developed worldwide involves employing a lot of different people to do a lot of different kinds of tasks. And this is not happening within any given one country. This is actually happening across the world. And so a lot of the decisions that get made in terms of how AI is pursued actually have impact on workers everywhere. And I think it's really important to point out that in terms of AI research, so I've been an AI researcher. I don't know when you officially become an AI researcher. There's no ceremony for that. But for me, my first research paper was in 1989, and I went to a conference, and it was a global conference in the sense that there were researchers from all over the world. And so my entire career in AI has been interacting with not just folks in the US doing AI research, but folks everywhere in the world doing AI research. It's a global academic community. And I think recognizing that we're not making these unilateral decisions. When we're talking about AI research, we need to be working together as a team. That to me is the impetus for doing something like this. And so I think this is kind of our offering out into the world saying, this is how we're thinking about this. This is how we'd like to work with you. Let's get this conversation going.
Special thanks to everyone who took time out of their schedules to join us and share stories about their amazing breakthroughs in 2024. And our thanks to you for listening. For the Discovery Files, I'm Nate Potker. You can watch video versions of these conversations on our YouTube channel by searching at NSF Science. Please subscribe wherever you get podcasts, and if you like our program, share with a friend and consider leaving a review. Discover how the U.S. National Science Foundation is advancing research at nsf.gov.